In 2019, a researcher at the University of Vienna carefully drilled into the petrous bone of a skull recovered from a necropolis just outside Rome. The petrous bone is the densest bone in the human body. It sits deep behind the ear, and because it is so hard, it preserves DNA far longer than anything else in a skeleton. Sometimes for thousands of years, in conditions that would destroy every other biological trace. The powder that came out of that tiny fragment would be processed, amplified, sequenced, and compared against thousands of other genomes, ancient and modern. And when the results came back, they told a story that contradicted almost everything the field had assumed about who lived in Rome, and when and where they came from. This is not a story about the Etruscans, or the Lombards, or the Romans. It is a story about layer after layer of people arriving on a peninsula shaped like a boot dangling into the Mediterranean, each wave overwriting part of the genetic record and leaving traces that would remain invisible for millennia. 5 major demographic upheavals, 12,000 years, all compressed into a genome you can read from a speck of bone dust. The question is not whether Italians are who they think they are. The question is whether anyone could have predicted what that dust actually contains. To understand what the genetics revealed, you have to understand what the consensus looked like before anyone had sequenced a single ancient Italian genome. For most of the 20th century, the story of Italy was told through language, pottery, and written records. Romans spoke Latin, which was Indo-European. The Etruscans spoke something else entirely, a language that linguists still cannot fully decipher despite decades of trying. The Greeks colonized the south, founding cities like Syracuse, Taranto, and Naples. The Gauls settled the Po Valley in the north, and after the fall of Rome, the Lombards and other Germanic peoples invaded from beyond the Alps. That much was not controversial. But the assumption underneath all of it was that modern Italians were, more or less, the genetic descendants of the Romans who built the roads and the aqueducts. That the deep biological identity of the peninsula had been set during antiquity, and everything since. The Lombard conquest, the Norman invasion of Sicily, the Arab Emirate, was cultural overlay on top of a stable genetic foundation. This assumption felt unshakable. Italy's regional diversity, the visible differences between a Neapolitan and a Milanese, were understood as ancient tribal echoes stretching back to the Iron Age. Northern Italians descended from Gauls and Lombards, Southern Italians from Greeks and native Italic peoples. Sardinians were a world apart, isolated on their island. The narrative was clean, it matched the archaeology, it made intuitive sense. And nobody had the technology to test whether it was actually true. That last part is the mechanism that allowed the assumption to survive unchallenged. Before ancient DNA extraction became reliable in the mid-2010s, population genetics could only work with modern samples. You took blood from living Italians, compared marker frequencies, and tried to infer backward through time. The problem is that inference cannot distinguish between two very different histories that leave similar modern signatures. A population that stayed put for 3,000 years and one that was replaced twice but landed in a similar genetic range look nearly identical when all you have is the endpoint. Mitochondrial DNA studies in the early 2000s compounded the confusion rather than resolving it. A study published in 2007 compared mtDNA from modern Tuscans to Anatolian populations and found a match, which seemed to confirm Herodotus, the ancient Greek historian who claimed the Etruscans had migrated from Lydia in modern Turkey. That finding rippled through popular science media. The Etruscans were Anatolian immigrants. The father of history was vindicated. Case closed. Except it was not closed. Mitochondrial DNA traces only the maternal line, mother to daughter to granddaughter. One thin thread of ancestry, while ignoring everything carried by fathers and missing the vast majority of the genome that actually defines a population's profile. Using mtDNA to reconstruct population history is like trying to understand a city's demographics by interviewing one person per household and only asking about their mother's side. You might catch a signal, but you are missing almost the entire picture. The tool that finally broke the problem open was whole genome sequencing of ancient DNA, pulled from those petrous bones lodged behind the inner ear. Because the petrous portion of the temporal bone is so extraordinarily dense, it can preserve usable genetic material in remains that are 8, 10, 12,000 years old, in Mediterranean climates that would normally degrade DNA within centuries. When researchers began applying this method systematically across the Italian peninsula, the clean story disintegrated. Margaret Antonio and Jonathan Pritchard at Stanford led the study that redefined the field. Published in the journal Science in November of 2019, it presented 127 ancient genomes from 29 archaeological sites in and around Rome. The time depth was unprecedented for a single region. 12,000 years of continuous human habitation sampled and sequenced. And the results did not show a stable population, gently absorbing newcomers over time. They showed a series of sharp genetic ruptures, each one transforming the biological composition of the peninsula's inhabitants. The first rupture came around 8,000 years ago, when Neolithic farmers originating from Anatolia and the Near East arrived and substantially replaced the Mesolithic hunter-gatherers who had inhabited Italy since the end of the last ice age. Those hunter-gatherers had been there for a very long time. Modern humans reached the Italian peninsula roughly 45,000 years ago, and during the last glacial maximum, when ice sheets covered much of northern Europe, Italy served as one of the refugia where small populations survived the cold and later re-expanded northward as temperatures rose. The farming transition was not gentle. Across Europe, it involved a near-total replacement of indigenous foragers by incoming agricultural populations. In Italy, the earliest farming sites appear in Puglia on the southeastern coast, and from there, the agricultural frontier spread both northward along the Adriatic and westward toward the Tyrrhenian coast. Within a few centuries, the genetic signature of the original hunter-gatherers was reduced to a minority component, typically around 15 to 25% of the genome. But this Neolithic foundation persisted most purely in one place: Sardinia. The island, difficult to reach by sea and far enough from the mainland to avoid the casual migration that reshuffled coastal populations, was largely bypassed by subsequent demographic events. Modern Sardinians remain, to this day, the closest living Europeans to those original Neolithic farmers. A genetic time capsule surrounded by blue water. The second rupture arrived during the Bronze Age, roughly 5,000 years ago, when populations carrying ancestry from the Pontic-Caspian steppe moved into Europe and, eventually, into Italy. Saupe and colleagues confirmed in their 2021 study, published in Current Biology, that this steppe-related ancestry reached northern Italy by approximately 2000 BCE and penetrated into central Italy by 1600 BCE. The vehicle was the Bell Beaker Cultural Complex, a widespread archaeological phenomenon associated with distinctive pottery, metalwork, and, as ancient DNA now reveals, a specific genetic profile. By the time Rome was traditionally founded in 753 BCE, Central Italians carried roughly 30% steppe-related ancestry. They had come to genetically resemble other Iron Age Mediterranean populations. Two massive demographic events in 4,000 years, and the population had already been overwritten twice. Now, here is where it gets genuinely strange, and where the clean narrative that Italians are simply descendants of Romans starts to look not just incomplete, but fundamentally misleading. In 2021, Cosimo Post at the University of Tübingen and an international team spanning Germany, Italy, the United States, Denmark, and the United Kingdom analyzed 82 ancient genomes from 12 archaeological sites across Etruria and southern Italy. The time range stretched from 800 BCE to 1000 CE, almost 2 millennia of burial evidence. They were looking for the genetic signature of the Etruscans, half expecting to find traces of the Anatolian migration that Herodotus had described and that the mtDNA study seemed to support. They found nothing. Zero signal of recent Anatolian admixture. No Eastern Mediterranean ancestry that was not already present in other Italian populations of the same period. The Etruscans were genetically indistinguishable from the Latins living in nearby Rome. Same proportion of steppe ancestry. Same Neolithic farmer foundation. Same profile. Two civilizations with radically different languages, distinct religious practices, different art, different funerary customs, and virtually identical DNA. Posch told Science magazine directly that the Etruscans look indistinguishable from their Latin neighbors. The Herodotus theory was not just weakened, it was demolished at the genomic level. And the 2007 mtDNA study that seemed to confirm it? That result now looks like a textbook example of what happens when you build a grand narrative on one thin strand of evidence and assume it represents the whole rope. If you have found these genetic layers as revealing as I do, and you want to see what happens when we apply the same methods to the other side of the Adriatic, subscribing tells us there is an audience for this kind of deep research. We are building a follow-up that traces how steppe DNA moved differently through the Balkans versus the Italian peninsula, and it gets even more tangled than this. But the Etruscan result was not the most disruptive finding in these studies. That distinction belongs to what happened next in the genetic timeline. During the Roman Imperial period, beginning around the 1st century CE, the genetic profile of people buried in and around Rome underwent a transformation so dramatic that it registered as one of the largest ancestry shifts documented in European history outside of the original Neolithic transition. The ancestry composition of Central Italian populations swung heavily toward the Eastern Mediterranean. Posch's team quantified it at approximately 50% admixture with Eastern Mediterranean populations. Antonio's study independently captured the same signal. People interred in Imperial-era Rome looked, genetically, less like their own Iron Age predecessors, and more like contemporaries living in Syria, Lebanon, and Western Anatolia. The mechanism was the empire itself. At its peak, Rome controlled territory from Britain to Mesopotamia, governing roughly 70 million inhabitants. Slaves poured into the capital from the eastern provinces. So did freed people, merchants, soldiers completing their service, administrators, and craftsmen seeking opportunity in the largest city in the Western world. The Eastern Mediterranean was far more densely populated than Gaul or Hispania or Britannia. The net demographic flow ran westward toward Rome. And this was not a trickle spanning centuries. The genetic data suggests it was rapid enough to reshape the city's ancestry profile within a few generations. Rome did not just conquer the East moved to Rome. A 2025 study published in Genome Biology and Evolution by researchers at Oxford found that this Eastern Mediterranean genetic signal appeared in Central Italian individuals dated to the late Republican period, roughly 200 years before the empire formally began under Augustus. The shift was already underway during the Punic Wars, the conquest of Greece, the annexation of eastern provinces. The genetics were tracking military and economic expansion more precisely than any surviving historical text. 3 proof beats lock this transformation into place. First, what the evidence shows. The Eastern Mediterranean ancestry shift appears simultaneously in multiple independent burial sites in and around Rome, in Etruscan-era necropolises from the same period, and in individuals recovered from sites outside the city entirely. It is not a fluke of one cemetery or one study. It is replicated across research teams using different sequencing platforms, different reference populations, and different statistical models. Second, what it rules out. The consistency rules out contamination, modern admixture, and sampling bias. It also rules out the idea that Imperial Rome remained genetically Roman in any meaningful Iron Age sense. By the height of the empire, the residents of the capital were as genetically close to populations in the Levant and Anatolia as they were to their own predecessors from 500 years earlier. The city that built the Colosseum and codified Roman law was, in biological terms, a Near Eastern city as much as a European one. Third, what it implies. It implies that the genetic identity of a civilization is not fixed by its founders. The Romans constructed institutions that endured for millennia. But the people who lived under those institutions were in constant flux. Within 300 years, migration reshaped the population so thoroughly that the genomic distance between an Imperial Roman and an Iron Age Roman was comparable to the distance between modern populations in different countries. But the story does not end with the empire. It has one more turn. After Rome fell, Germanic populations swept into the peninsula. The most significant were the Lombards, who crossed from Pannonia into Italy beginning in 568 CE. A 2018 study published in Nature Communications analyzing Lombard burials in both Hungary and Italy found strong genetic similarities between the two groups, confirming this was a genuine folk migration, families included, not merely a military conquest. Lombard males carried Y chromosome lineages characteristic of Northern and Central European Germanic populations, particularly subclades of haplogroup R1b and I2A2A1. The Lombard migration did not reach every corner of Italy. They controlled Lombardy, Tuscany, parts of the center, and the duchies of Spoleto and Benevento in the south. But they never took Sardinia, most of Sicily, Calabria, or coastal Puglia, regions that remained under Byzantine governance. This uneven coverage created a genetic fault line that persists to this day. Northern and Central Italy absorbed a Northern European genetic component that partially diluted the Imperial-era Eastern Mediterranean signal. Southern Italy, under Byzantine control, retained more of that Eastern Mediterranean and Greek colonial ancestry. And the Greek colonial ancestry was substantial. Beginning in the 8th century BCE, Greek settlers established colonies along the coasts of southern Italy and Sicily, so extensive that the Romans later called the entire region Magna Graecia, Greater Greece. Syracuse became one of the largest cities in the ancient world. Taranto, Naples, Reggio Calabria, all began as Greek foundations. These were not trading posts. They were full-scale settlements with temples, theaters, and governing institutions, and their populations intermarried with local Italic peoples over centuries. Recent DNA analysis has demonstrated that this Greek colonization left a significant and lasting genetic signature. Modern people from southern Italy and Sicily carry measurably more Greek-related ancestry than their compatriots to the north, a signal that has survived not just the Roman period, but also the Arab Emirate in Sicily, the Norman conquest, and 5 centuries of Spanish rule. And this is exactly what Reevaluation and colleagues found when they mapped the genetic structure of 1,616 modern Italians from all 20 administrative regions in their 2019 study in Science Advances. Italy contains the largest degree of genetic population structure detected in any single European country. 3 distinct clusters emerged: Northern Italy, including Tuscany; Southern Italy, including Sicily and the central southern regions; and Sardinia, standing entirely apart. Southern Italians are genetically closest to modern Greeks, a legacy not only of ancient Magna Graecia, but of centuries of shared Byzantine governance and Eastern Mediterranean connectivity. Northern Italians cluster with Spaniards and Southern French, reflecting the deeper penetration of steppe ancestry and the later addition of Germanic elements. Sardinians are the outliers of all outliers, preserving a genetic profile closer to Stone Age farmers than to anyone else alive in Europe today. The genetic distance between a Milanese and a Sicilian is modest in absolute terms, an FST of about 0.0013, roughly equivalent to the distance between northern and southern Germans. But in the Italian context, that number maps precisely onto 12,000 years of differential migration history, layer by layer, region by region, conquest by conquest. Not everyone in the field agrees on the interpretation. Some researchers argue for continuity. The basic framework of Italian genetics, they suggest, was established by the Bronze Age, and everything since has been adjustment at the margins. The Eastern Mediterranean influx during the Empire was concentrated in Rome and its immediate surroundings. The Lombards were a thin ruling elite, not a mass migration. The deep structure endured. I find that position increasingly difficult to defend against the actual numbers. 50% Eastern Mediterranean admixture in Imperial-era Central Italy is not an adjustment at the margins. That is a population-scale replacement event. If 50% of your genome is now from a different part of the Mediterranean than it was 4 centuries earlier, the word continuity is doing a lot of heavy lifting. And the Lombard migration, while smaller in scale, has now been confirmed through ancient DNA as a genuine population movement with families in tow, not just a change of rulers. Calling either event marginal requires looking past the very data that revealed them. The 2024 study on the Piscine culture, published in Genome Biology, adds another layer to this question. Researchers sequenced over 50 individuals from Adriatic Central Italy spanning more than 1,000 years, from the Iron Age through late antiquity. Despite the Piscenes having a distinct culture from the Etruscans and the Latins, their genetic profile was remarkably similar to both. The Iron Age Italian peninsula, it turns out, was culturally fragmented but genetically cohesive. Multiple identities, one biological substrate. That finding only makes the Imperial-era disruption more striking. A genetic unity that survived centuries of cultural competition was shattered within a few hundred years of imperial expansion. And then there is the Etruscan paradox, which refuses to resolve cleanly. If the Etruscans carried steppe ancestry, and steppe ancestry is associated across the rest of Europe with the spread of Indo-European languages, why did the Etruscans speak a language that was not Indo-European? David Caramelli at the University of Florence offered one explanation. The Etruscan language predated the steppe arrivals and was simply resilient enough to survive the genetic turnover. New arrivals assimilated into the existing Etruscan culture rather than overwriting it. Genes changed, language persisted. That is plausible, even elegant. But it makes the uncomfortable point extremely sharp. The assumption that genetic ancestry predicts cultural identity, the assumption that underwrote decades of population history, is precisely the kind of shortcut that produces beautiful narratives and wrong conclusions. What remains genuinely unknown is how uniform the Imperial-era genetic shift was beyond Rome and Etruria. Most ancient DNA from Italy comes from a handful of well-excavated sites in Lazio and Tuscany. There are far fewer sequenced genomes from Calabria, Puglia, Campania, or the deep interior of Sicily. If the Eastern Mediterranean influx was concentrated in urban centers along trade routes, the story is one of localized cosmopolitanism. If it was peninsula-wide, it rewrites even more than we currently understand. A 2025 study on Bronze Age remains from Grotta della Monica in Calabria, published in Communications Biology, found that even in the Bronze Age, genetic trajectories in the Deep South diverged from those in the North. Calabrian Bronze Age individuals showed closer affinity to Sicilian populations than to contemporaneous groups on the Italian mainland, and they lacked the Eastern genetic influences already present in Sicily at the same time. This suggests that even long before Rome, the peninsula was not a single genetic unit, but a patchwork of micro-regions, each with its own specific pattern of contact and isolation, shaped by mountains, river valleys, and access to the sea. Stand at a bus stop in Rome today, and you are standing on 12,000 years of genetic layering. hunter-gatherers who survived the Ice Age in Mediterranean refugia, replaced by Anatolian farmers who brought wheat and sheep and a completely different way of life. Farmers blended with steppe pastoralists from the grasslands north of the Black Sea, people who rode horses and carried bronze and spoke the ancestor of Latin itself. Iron Age populations absorbing Greek colonists in the south and Celtic groups in the Po Valley. Then the Empire, drawing in DNA from Syria and Lebanon and Egypt and North Africa, rewriting the genome of the capital within a few hundred years. Then collapse, and the Lombards arriving from beyond the Alps. Then the Normans in Sicily, the Arabs before them, the Byzantines holding on in the coastal south. Every layer still present, every layer still measurable. And the technology that reads them, the ability to extract a sequence from a fragment of bone behind the ear of someone who died 2,000 years ago, has existed for less than a decade. The next petrous bone pulled from an Italian necropolis could rewrite this story again. That is not speculation. At this point, it is the pattern.